Hello, can you hear me? Okay. Hi, I'm Felicia. So I'm from the Institute of Molecular and Cell Biology A*, and today I'll be presenting my team's work titled An Interactive Tool for Analyzing and Visualizing Cellular Data Generated from Tissue-Based Multiplexed Immunofluorescence Images. So just a quick introduction as to what multiplex immunofluorescence images are, or MIF for short. So MIF is actually a technique whereby researchers can use to stain and to visualize protein biomarkers of interest using antibody detection on the same tissue section. So here I have two samples from two different patients, and in both samples we can observe that there are immune clusters as well as tumor cells, but only in the sample on the left do we see immune cell infiltrates. And usually the presence of immune cell infiltrates means that it will impact the patient prognosis as well, so it's pretty important. So I just want to give a quick demonstration as to how important or how powerful a tool that MIF is in order to do visual interpretation as well as qualitative analysis. So now I'll talk about the quantitative analysis portion, and it comes with its own set of challenges to analyze these MIF images. So MIF is actually part of it's a technique of tissue-based spatial proteomics, and these images have a very high plex, meaning that we have a lot of target protein biomarkers. So we have 40 in our case, and usually when we run the samples, we have a batch of 4 samples per batch, and usually we are able to stain and to get the images out within 36 hours, and each sample has at least 0.3 million cells. And we also rely heavily on image-based analysis. So essentially what we're doing is to transform high-resolution whole slide image data to cell-level data, and that generates a lot of information at the single-cell resolution. And we also have information at the subcellular level, so like the nucleus, the cytoplasm, the cell membrane. And because this is spatial proteomics after all, right? So it involves the spatial location of each and every cell as well. And overall, the data is very high-dimensional. So it has, so all these factors combined, it's quite challenging for, let's say, a non-bioinformatician to analyze. So I'll just bring you through our current workflow. So what we do is we do staining and image acquisition, and this is all automated in a machine. Essentially what the machine does is to stain 2 antibodies, take the image of the sample, remove these 2 antibodies, and then stain the next set of antibodies. So this is done in a sequential manner, such that at the end we get a stacked image with the 40 protein biomarkers, as well as a nuclear counterstain called DAPI. Next, we do cell segmentation using the image-based image analysis software. So in our case, we use a pre-trained AI algorithm to do the cell segmentation, and then we do the thresholding of each and every protein biomarker so that we define the positivity in a binary fashion, which then helps us to do cell phenotyping. And we export all these cell level data as an Excel sheet, so that includes the XY coordinates of each and every cell, as well as the stain intensity values. And we then pass it to the bioinformatician to analyze, and the results are then given to the biologist to interpret. However, sometimes the results that we get could be not so straightforward to understand, or perhaps more troubleshooting is required. So usually there's a lot of discussions between the bioinformatician and the biologist, going back and forth to try and make sense of the data, to try and understand the data and make sure that it matches back to the clinical data. So these are some of the bottlenecks within our current workflow. So firstly, images are acquired really quickly. Like I mentioned, every batch run that we have, we obtain the images of 4 samples within 36 hours. So that's 1.5 days. So that's really fast, and sometimes analysis can be very complex and it takes time to complete. So for our workflow, our analysis is done in a 2-part manner. So the first part is done in an image-based analysis software, and then followed by the cell level data being done in programming applications, for example. And also, like in the previous slide, I mentioned that a lot of discussions are required between the 2 parties involved in the whole analysis pipeline, so that takes a lot of time as well. So in order to overcome all these issues, our team decided to utilize the Shiny app. So this is a code-free, interactive, user-friendly app where researchers can use to analyze the data, to make quick overview of the data, and you can even run, like, let's say, a pilot run to understand your data. And this part, your analysis pipeline can be semi-automated as well. The video playing? Yes. Okay. So here is a video demonstration of our Shiny app. This is done by our fabulous team member Yan Chun. So essentially, users can just upload their Excel sheets, and then you can choose to subsample your data if you want. So for this particular video here, we chose to subsample 50,000 cells, and we have 4 different visualization features. So we have the expression heat map, the co-expression heat map, the dimension reduction plots, as well as the spatial visualization plot. And in the interface is very simple to use. It's very intuitive. You can just select the different drop-down options that are available. And because of the reactive programming, the images come up pretty fast as well. Yeah, so there we go. And then just to give you some insight as to each of the features that we have. So the first one is the marker expression heat map. What it does is that it will visualize the level of protein biomarker expression of each cluster. And for this particular example here, we use the full data set of 300,000 cells. And users can choose to do 2 types of scaling options, either row or column. And you can also choose to do 3 types of k-value selection, so either 30, 50, or 150. So these are the nearest neighbors to do the phenograph clustering. And for this data set, we identified 26 classes using the phenograph unsupervised clustering. And from this, we do the manual cell type annotation for each and every cluster so that we identify what cell types are within each cluster. So the second one are the dimension reduction plots. So I think everyone's very familiar with this, that we can preserve spatial relationship between data points in a lower dimensional space. So users can select between either the t-SNE or the UMAP, and similarly, you can choose one of the 3 k values here. So for this particular sample, we identified 3 distinct clusters. So cluster 9 contains macrophages, cluster 10 we identified as NK cells and monocytes, and then cluster 25, which is identified to have granulocytes. So the next feature is really interesting. What we do is we take the different phenograph clusters on the left, and then we plot them back into the 2D tissue space. So the image on the right actually shows how the real tissue sample looks like. Yeah, so the annotated clusters are reconstructed to form the 2D tissue image to visualize localization patterns of cell phenotypes. And for this particular sample, we found that the tumor infiltrating neutrophils, which is cluster 22 in orange right here, they are found within the tumor nest, which is cluster 26 in light blue. Where's my pointer? Here, all these blue areas. Yeah, so the image on the left shows the hematoxin and eosin image. So H&E is a very common stain used in histology to identify or to read the global tissue structure and landscape. So from here we can see that the just earlier I mentioned that cluster 26 contains the tumor cells, which is here. All these, they're actually the same reflected here. All these are the tumor cells. And this is the original MIF image that we have. Similarly, we can tell which are the tumor cells. And then if you focus on this cluster 10 right here in mid blue, these actually correspond to this group of cells here. So because of our analysis pipeline, we managed to identify that these cluster of cells are NK cells as well as monocytes. Yeah, so the final feature that we have is the marker co-expression heat map, where we use Spearman's correlation to do this. And it displays the frequency of protein co-expression on cells as a heat map. So for this particular sample, we identified that there are high co-expressions of immune cell markers, in particular the B cell marker CD20 and CD21, and we also identified the presence of inflammatory dendritic cells. Yeah. So this is the workflow that I showed earlier. Now with the introduction of our Shiny app visualization analysis, the biologists can first do interpretation as well as quality check of their results. So for example, usually our patient cohorts can be quite big. Let's say we have 60 patients and we're not sure if the data we're getting is going in the right direction or it matches back to the clinical analysis, clinical data. So what we can do is to run a pilot run first of, let's say, 5 samples, and then we test it out and then make sure that it makes sense, the data makes sense and it matches back, and then we can run the whole cohort and pass the whole cohort to the bioinformatician downstream to do further analysis. So here are the benefits to users if they use our Shiny app. So firstly, no prior coding experience is required. It's very interactive, user-friendly, the interface is very intuitive to use, and because we use open source analysis framework, our users can always seek help or support from the online community who are very supportive in these efforts. And of course, is to because biologists can understand the analysis results more, we will have more efficient collaboration and discussions between the both parties of biologists and bioinformaticians. And we can also do quick evaluation of our data and to validate experimental hypotheses. And we can also choose to do subsampling of our data just to have a quick check of whether it makes sense or not. Yeah, so here are some of the future works that we have in mind. So we want to extend the use of our app to other multiplex modalities like imaging mass cytometry. And then we also want to include more visualization features. So some of the manual cell annotations that we did, we want to import them back into the phenograph cluster so that it's easy to refer to. And we also want to allow upload of other data formats such as CSV files and SIRAD objects. And we also want to implement a function to allow users to export visualizations in high resolution so that they can use it for their manuscripts. Yeah, so thank you, and I want to thank this list of people right here because this wouldn't have happened without the support. Yeah, thank you.
Thank you, Felicia. We have time for some questions.
Hello, thank you. Nice work. I saw that you have multiple examples of 2D image analysis. Did you try to do a 3D analysis and cell segmentation and annotation, but in a 3D space?
We have yet to try that, but I think that will be really, really cool to do because our images that we obtain is also in 2D. Yeah, so if we want to work on the 3D plotting, right, we will have to start off with perhaps a 3D modeling of our sample as well. But currently, our images are all in 2D. Yeah.
Thank you.
Okay, thank you.
So you mentioned, if I understand correctly, that you were using this for sort of multiple patients. So I'm wondering how you account for sort of batch effects and the different stainings between different patients, and then if you can actually correspond the same cell types in different actual people.
So currently, because each and every tissue has autofluorescence, so what the machine does is to first take the autofluorescence channel, and then it subtracts that from the staining. So it always makes sure that across the different batches, it will be pretty consistent. Yeah.
Okay, so you use like autofluorescence as a standard candle to then do the baselining.
Yes, correct.
Okay, thank you.
So you may have mentioned, but is your Shiny app available, publicly available?
Currently, it's not, but we should try to push for it. Yeah.
Okay, thank you.
Hi Felicia, really nice talk. I think it's more like a non-technical question. Have you tried to time the entire process and how fast it speeds up the whole process really?
Let me think. So when I ran the full data set, that 300,000 cells, right? I think it took about 40 minutes for the whole process for all like the 4 different plots to be generated. But the one that I realized took a lot of time was the T-SNE. Yeah.
Okay, no, because I think my question is really like on the workflow level, without the Shiny app, how long would that take? And then, how long would the whole process take with the Shiny app? I think that would be a really interesting thing to test.
Yeah, I think we should go and measure that as well, but definitely faster.
Okay, thank you.
Any other questions? Do you also have a plan to release it as a sort of downloadable package? Because if it's only online, then perhaps sometimes there may be some jam if there's a long queue.
Okay, so the thing is, we're also hoping that non-bioinformaticians will use. So I'm not sure if they will need to download package. Will they know how to incorporate it? Yeah, so that's why we try to make it as like a website, so that they, or rather an app, so that they can easily just plug in the cell level data that they have, and then plots are generated.